Today we're going to learn about epigenetics inheritance in breast cancer.
Epigenetics has been defined and today is generally accepted as the study of changes in gene function that are mitotically and/or meiotically heritable, and that do not entail a change in DNA sequence. The epigenetic modifications described in current literature generally comprise histone variants, post-translational modifications of amino acids on the amino terminal tail of histones, and covalent modifications of DNA bases. Epigenetics is the study of heritable phenotype changes that do not involve alterations in the DNA sequence.
DNA methylation in breast cancer. Emma, could you please explain more about how DNA methylation causes breast cancer?
DNA methylation is the covalent modification that take place at the 5' end of the CpG dinucleotide of the cytosine ring and with S-adenosylmethionine as its methyl donor, which located in the promoter region of genes and to generate 5-methylcytosine. This reaction is catalyzed by the DNA methyltransferase, DNMT, family such as DNMT1, DNMT3A and DNMT3B. DNMT family is vital important to maintain genome stability. DNMT3A and DNMT3B are two closely related de novo methyltransferase, which involved in methylation during embryonic development. An activation of both genes caused a complete failure to establish genome-wide methylation. Moreover, an activation of either gene alone may lead to hypomethylation at a subset of sequences in the genome, but most sequences are unaffected. DNMT3A is required for the methylation of imprinted genes in germ cells, while DNMT3B has an irreplaceable function in the methylation of centromeric minor satellite repeats. So, genomic targets can be catalyzed by these two enzymes. For DNMT1, methylate hemiethylated sites generate during DNA replication in order to maintain the methylation status and aberrant gene silencing in human cancer cells. DNMT1 is the most abundant DNMT in adult cells. The covalent modification is inherited through cell division. By blocking the combination of a transcription factor or its binding sites will inhibit the activity of gene transcription. The diagram shows a normal DNA sequence in gene transcription, and cancer gene transcription. There are two contradictory changes in DNA methylation patterns. These are observed in breast cancer. Regional hypomethylation of specific genes and global hypomethylation. Regional hypomethylation is brought about by specific regional changes in chromatin structure. Hypomethylation silence growth regulatory genes, resulting in uncontrolled growth. For global demethylation, it is caused by a general increase in demethylation activity and hypomethylation. This leads to activation of genes which are required for metastasis and invasion. Global hypomethylation is frequently reported in breast tumors, but the number of hypomethylated genes is relatively small. DNA hypomethylation of FEN1, BGSC1, PLAU, IGF2 and CDH3 has been detected in breast cancer cells, although there had two types of DNA methylation. There have more than 100 genes that considered to be regional hypomethylated in breast cancer. When the cancer development, many of these genes are hypomethylated at their CpG island containing promoters to inactivate with their expression, by changing open euchromatic structure in order to compact heterochromatic structure. For example, CCND2 and P16INC4A or CDKN2A function as important regulators in the cell cycle. They are usually found to be methylated in breast cancer. APC, Twist and HOXA5, which play an important key roles in apoptosis, are silenced due to DNA hypermethylation, while ER-alpha and PR, which are critical in hormone regulation. Regions which consists of high density of CpG is known as CpG island. They are found in regulatory regions such as promoter and lead to inactivation of transcription. CpG islands are much less common in tissue-specific genes. This DNA hypermethylation plays an important role in breast carcinogenesis. It interacts cooperatively and synergistically with other genetic alterations to promote the development of breast cancer. Besides, DNA methylation inhibitors activate silenced tumor suppressor genes. As the result in arrest of tumor growth and are now being tested as candidate anti-cancer drugs. Epigenetics is the study of heritable phenotype changes that do not involve alterations in the DNA sequence. Epigenetics most often denotes changes that affect gene activity and expression, but can also be used to describe any heritable phenotypic change. Such effects on cellular and physiological phenotypic traits may result from external or environmental factors.
DNA methylation plays a crucial part in breast cancer by mechanism of gene silencing for cancer prevention and therapy. Why not check out our new video on YouTube about breast cancer awareness? Thanks for watching.
We hope to see you soon in our next video. Goodbye.
Goodbye.